Press the bell icon on the YouTube app and never miss another update.
Hi everyone, welcome back. In the last video we discussed the spermatogenesis process. So in this session I am going to discuss a detailed structure of the human sperm. Okay. So you already know about the human sperm. It is a haploid, motile, flagellate structure. Okay. So the human sperm is haploid. Haploid means a single set of chromosomes. Motile, capable of movement. So it is having a flagella. For motility, it has developed a structure called the flagella. The flagella helps in the motility of the sperm. And it is microscopic in nature. Microscopic. So it measures about 60 micrometer in length and nearly 3.5 micrometer in breadth. So this is what the size of the human sperm. See, human males they produce two different types of sperms. Two types of sperms. So two types of sperms in the sense you just recall the process of spermatogenesis. So in spermatogenesis process what? The primary spermatocyte, it undergoes a first meiosis and gives rise to two daughter cells. These two daughter cells are called as secondary spermatocytes. Again the two daughter cells, secondary spermatocytes undergoes a meiosis second and gives rise to four haploid spermatids. So these spermatids undergo changes, morphological and physiological changes that results in the formation of four functional sperms. So this is what the spermatogenesis. See here, a primary spermatocyte, no? So what is the ploid of primary spermatocyte? 2n. Right. So what is the number of chromosomes then? Diploid set of chromosomes. So in human beings it is 46. Out of 46, 44 are autosomes and remaining two chromosomes, they are called as what? The sex chromosomes. So what are the sex chromosomes in males? X and Y. So this primary oocyte undergoes meiosis first, that gives rise to two daughter cells. So the resulting daughter cells are haploid with 23 number of chromosomes. So each daughter cell having 23 number of chromosomes. Out of 23 chromosomes, the 22 are autosomes and the 1 plus 1 chromosome, either X chromosome or Y chromosome. Okay. So if the daughter cell, this daughter cell receives X chromosome. So what is the chromosome number here is 23. In that 22 are autosomes and X chromosome, and this daughter cell is having 22 autosomes and a Y chromosome. Okay. So when these two daughter cells again undergo meiosis second, four daughter cells are produced. Okay. So half of the cells having receives the X chromosome and remaining half of the daughter cells will receive a Y chromosome. So of the sperms, 4 sperms are formed here, no? Out of 4 sperms, the 2 sperms are having X chromosome and the 2 sperms are having Y chromosome. Okay. So here the sex of the baby is determined based on the type of the sperm fuses with the egg. Okay. So if it fuses with the X carrying chromosome, X carrying sperm fuses with the egg, that results in a female baby, and whereas the Y chromosome carrying sperm fuses with the egg, that results in a male baby. Okay, that you will discuss in detail in the genetics chapter. But what is important here is the males will produce two types of sperms. Half of the sperms having X chromosome and remaining half of the sperms are having Y chromosome. Okay. Now let us come to the structure of the typical human sperm. So this human sperm diagram is very important. You need to learn the diagram. A typical human sperm having 4 distinct portions. That is head region, head, neck, middle piece and the tail. So what are the 4 distinct regions of the sperm? Head, neck, middle piece and the tail. Okay, so the anterior part of the sperm is called as, it's a knob-like structure. This part is called as what? The head. So the head includes 2 important structures. One is the nucleus and another one is a smaller, in the smaller anterior part it is having, in the anterior part it is having a small acrosome and the posterior part it is having a nucleus. Okay, so what is present in the head region? Towards anterior side acrosome, towards posterior side nucleus. Okay, so as I discussed in the last video, so when the primary spermatocyte completes meiosis, the resulting daughter cells, they are called as what? Spermatids. Okay, you can see the difference in the shape of the spermatids and the shape of the sperm. So they are spherical in shape. They do not have flagella for motility, okay, and they do not function like sperms also. But what are all the structures which are present in the spermatids? No, so they will undergo changes that finally results in a functional sperm. Okay, so the first thing that is going to change is the nucleus. So the nucleus, it becomes compact and it occupies the major portion of the sperm head. So here, the nucleus, the shape of the nucleus will also changes. It gets elongated and anteriorly it is pointed and posteriorly it is having a depression. So what is the function of nucleus? The nucleus consists of haploid set of chromosomes and it contains what? A genetic material. Okay, the major portion of the head region is having the nucleus. And here you can see the presence of a cap-like structure above the anterior region of the nucleus. So this is called as what? The acrosome. Actually this acrosome is a modified Golgi complex of the spermatid. Okay. So what Golgi complex present in the spermatid, it changes to form an acrosome and it will form a cap-like structure over the nucleus. This is called as what? The acrosome. So here the acrosome is a modified Golgi complex. Okay, what is the function of this acrosome? So in the acrosome there are certain enzymes are present. So those enzymes, they are collectively, they are called as sperm lysins. They are collectively called as what? Sperm lysins. See, what is the function of sperm lysin? So they are helpful at the time of fertilization. See, at the time of fertilization, the sperm has to fuse with the egg, right? Okay, now. So at the time, the egg, it is covered by egg membranes. So the sperm lysine function is, they dissolve the egg membrane so that a sperm head can penetrate into the egg. Okay. So what is the function of sperm lysine then? The sperm lysine helps in the dissolving of, they digest the egg membranes so that the sperm head can penetrate the egg at the time of fertilization. Okay, then, so that is what the function of the acrosome. So in the head region, 2 important structures present. One is the nucleus, it contains the genetic material, and the acrosome, which contains sperm lysine enzyme. Okay, then the head is followed by a very short, narrow region of the sperm, that is called as what? The neck region. So, what is present in the neck region? See here is a depression. Okay, so in the depression lies the 2 centrioles what present in the spermatids, no? So they get arranged one after the other behind the nucleus. So one centriole will come to lie immediately below that what depression which is created in the nucleus. So the centriole which is located near to the nucleus, this is called as what? Proximal centriole. And one centriole which is away from the nucleus, this is called as what? The distal centriole. So both the centrioles play a very important role. What is the function of proximal centriole? Proximal centriole, it helps in the formation of spindle fibers. It helps in the formation of spindle fibers. at the time of the first mitotic division of the fertilized egg. Okay, so after fertilization zygote forms no, so the zygote undergoes cleavage no, mitotic cell division no, during the first mitotic division of the zygote, this proximal centriole helps in the formation of spindle fibers. Then this is followed by a distal centriole. See here, what is the function of distal centriole? So the distal centriole, it will give rise to the axial filament. Okay, so this is what the function of distal centriole. It will give rise to what? The axial filament. Okay, so the neck is followed by another long cylindrical little wider part of the sperm, that is called as what? The middle piece. The middle piece is having, in the center of the middle piece there is a presence of axial filament, which arising from the distal centriole. Okay, then the mitochondria what present in the spermatid no, it gets spirally coiled around the axial filament. So if it is a axial filament, the mitochondria, it gets spirally coiled around the axial filament. So this is called as what? The mitochondrial spiral. So this mitochondrial spiral is also called as Nebenker. What it is called? Nebenker. Okay. So this chamber, middle piece, it is also called as energy chamber. Why? Because the mitochondria, the mitochondria will provide energy for the movement of the tail, so that it helps in sperm motility, so that the sperm can reach the site of fertilization. So what is the function of mitochondria here? It provides energy for the movement of the tail. That helps in sperm motility. Okay, so that's why this region, middle piece, it is called as the energy chamber. Then after the middle piece, the next portion, the longest portion of the sperm, it is called as the tail. Okay, so of the four parts you see, no, head, neck, middle piece and tail, the longest part of the sperm is the tail. So what is the size of the sperm here? It measures about 60 micrometer in length. No, so here the tail alone measures about 50 micrometer in length. So it is the longest part of the tail. See, the tail is having in the center, it's also having the axial filament and the cytoplasm. So the what cytoplasm which is present in the spermatid, no, much of the cytoplasm will get lost. Okay, only a little amount of cytoplasm is left. So that cytoplasm gets arranged as a thin layer around the axial filament in the tail region. Okay. And again it is covered by a plasma membrane. See, actually the tail it is differentiated into two regions. The initial part of the tail, this is called as the main piece, and the later part it is called as the end piece. So the main piece is having a central axial filament surrounded by a thin layer of cytoplasm, and it is covered by a plasma membrane. Okay, so the plasma membrane will surround the entire part of the sperm, whole sperm, except in the region of end piece. Okay, so the main piece is followed by end piece. So in end piece there is a lack of cytoplasm and the plasma membrane. So okay, that's how the end piece and main piece gets differentiated. And here one more thing. So the posterior part of the nucleus and the neck region, and as well as the middle piece. Okay, so there is a presence of sheath that is called as Manchat. So Manchat is a temporary microtubular element that is formed during the spermiogenesis. So its function is, so it is believed to play a role in providing a shape to the nucleus. Okay, so that is what the function of Manchat. So the entire sperm, it is covered by a plasma membrane except in the region of end piece. Okay, so this is what the structure of human sperm. This diagram is very important. Okay, so it's having four portions: head, neck, middle piece and tail, and each and every part having its own role. Acrosome contains sperm lysins to dissolve the egg membrane. The nucleus, it contains a genetic material. Manchat provides shape to the nucleus. Proximal centriole, it helps in the formation of spindle fibers. So it is helpful at the time of first mitotic division of the zygote, and the distal centriole that helps that gives rise to the axial filament. and the mitochondria provide energy to the motility movement of the sperm, okay, and the flagellum or the tail helps in the motility of the sperm, okay. So this is what the structure of human sperm, okay. Now we will discuss the hormonal control of spermatogenesis. Hormonal control of spermatogenesis. See, once the males attain puberty, so many changes will occur in the body, and so many secondary sexual characteristics will develop, and they are able to produce gametes, so thereby they are able to reproduce, okay. So all these changes in males which occur at the puberty, it is all because of hormones. So which hormones will bring these changes in the males? Let us see. GnRH, gonadotropin releasing hormone. The second one is FSH. FSH is follicle stimulating hormone. One more is LH. LH is luteinizing hormone. In males, luteinizing hormone is also called as ICSH. And one more hormone, that is the testosterone. So testosterone is secreted by the interstitial cells or the Leydig cells of testis. Okay, so here the gonadotropin releasing hormone. Why it is called gonadotropin releasing hormone? Its function is this hormone is responsible for the release of gonadotropins. So which hormones are called gonadotropins? FSH and LH are called as gonadotropins. Why these are called gonadotropins? Because their target organs are the gonads, testis and the ovary. So that's why the FSH and LH are called gonadotropins. And one more hormone. So this gonadotropin releasing hormone is responsible for the release of gonadotropins. Okay, so that's why it's called gonadotropin releasing hormone. And one more hormone is testosterone. So the testosterone, it is necessary for the production of the sperm. So it stimulates spermatogenesis, and it is also responsible for the development of secondary sexual characteristics in males that we already discussed in our last classes. Okay, see here. The hormonal control of spermatogenesis. Gonadotropin releasing hormone. So this gonadotropin releasing hormone, so the spermatogenesis in human beings, the spermatogenesis process, it begins at puberty. Begins at puberty. Okay. And it will continue till death. So once the process begins with puberty, the males are able to produce the sperms throughout their life. So this spermatogenesis process is because of the increased secretion of gonadotropin releasing hormone. So this hormone is released by the hypothalamus. Hypothalamus. Right? So hypothalamus is a part of our brain. So once the males attain puberty, the hypothalamus increases the secretion of GnRH, gonadotropin releasing hormone. So this gonadotropin releasing hormone, the target gland for this hormone is the pituitary gland, particularly the anterior pituitary. So the gonadotropin releasing hormone will act on anterior pituitary, right? It act on anterior pituitary and stimulate the anterior pituitary to release gonadotropins. The gonadotropins are LH. It's also called ICSH. And one more is FSH. Okay, so both these hormones, gonadotropins, are released by the anterior pituitary under the influence of which hormone? Gonadotropin releasing hormone. Okay, now the target cells for these hormones in the testis. LH, the target cells for LH, Leydig cells. Where Leydig cells are present? They are also called as interstitial cells. No, they are present in the connective tissues of the testis. What is their role? Secretion of testosterone. The hormone secretion is under the influence of which hormone? Luteinizing hormone. Interstitial cell stimulating hormone, because it stimulates the interstitial cells or Leydig cells to secrete a testosterone hormone. Okay, so the hormone which is produced in response to LH causes the appearance of the development of secondary sexual characteristics in males, and also it is responsible for the production of the sperms. And FSH, mainly the target cells are Sertoli cells. So the FSH released in response to GnRH will act on Sertoli cells and stimulate the Sertoli cells to release certain factors that helps in spermiogenesis process. Apart from that, the Sertoli cells in response to FSH also releases Sertoli cells in response to FSH also releases androgen binding protein. What is the function of androgen binding protein? Androgen binding protein, its main function is it concentrates the testosterone in the seminiferous tubule. See here, this is the seminiferous tubule. Okay, testosterone, it is produced outside the seminiferous tubule by which cells? Leydig cells. So ABP is released by Sertoli cells present in the seminiferous tubules. So this ABP, what released by the Sertoli cells, it has a greater affinity for testosterone, so that the testosterone can bind to the androgen binding protein. So their that hormone concentration increases in the seminiferous tubule. Okay, so what is the function of Sertoli cell here? In response to FSH, the Sertoli cells will produce the ABP, androgen binding protein. The androgen binding protein increases the concentration of testosterone in the seminiferous tubule, right? And so testosterone is necessary for the production of the sperm. Okay, so this is how the LH stimulate Leydig cells. The Leydig cells will release testosterone, and FSH will act on Sertoli cells. The Sertoli cells will release some factors that helps in the process of spermiogenesis, and also releases androgen binding protein that concentrate the testosterone in the seminiferous tubule. Okay, Sertoli cells apart from FSH, apart from Sertoli cells, they also act on spermatogonial cells and stimulate the process of spermatogenesis. Okay, see spermatogenesis process, it is a continuous process. It is happening continuously. So because the germ cells undergo repeated mitotic division, produces more and more spermatogonia. Some spermatogonia will enter into that spermatogenesis process, and remaining spermatogonia again acts as a stem cells. They again undergo mitosis, produce more and more spermatogonia, and those spermatogonia will also carry out the spermatogenesis process. In this way, the sperms are being continuously produced in the testes. Okay, but the sperm formation is regulated by, so here for that to regulate the spermatogenesis process, it has a negative feedback mechanism. See, the increased concentration of testosterone the increased concentration of testosterone, it will give a negative feedback to hypothalamus. It will give negative feedback to hypothalamus. Okay, so thereby it reduces the release of gonadotropin releasing hormone, thereby the LH. And it is also have negative feedback to anterior pituitary. Okay, so thereby the secretion of that is LH hormone level comes down. So if LH hormone level reduces, the Leydig cells, they reduces what they secrete testosterone, no? The hormone production, the testosterone hormone production also gets reduced. Okay, then the increased that excessive spermatogenesis. Excess spermatogenesis causes the release of a hormone that is called inhibin from the Sertoli cells. So inhibin hormone, it is released by the Sertoli cells in response to excessive spermatogenesis. So this inhibin has a negative feedback to anterior pituitary. It has a negative feedback to anterior pituitary. So thereby the synthesis and the secretion of FSH hormone gets reduced. Okay, so once the FSH hormone gets reduced, the spermatogenesis process can be regulated. Spermatogenesis process is regulated. Okay, so this is what the hormonal control of spermatogenesis. Understood? All of you understood? Okay, thank you.